Okay.
Is that you?
That is me. Hello. Okay, so since it's only like 6 of us, it's gonna be pretty chill today. Alright, so the goal is to get through nervous and musculoskeletal. Did you guys finish that today?
We got like 4 slides away.
Okay, okay, cool. So last week with neuro stuff, we talked a lot about how we went over the normal phys. So we went over like the different parts of the brain, what they're all important for. So remember, the front part of your brain is very important for kind of motor and how you interact with the world is how I think about it. So you have this central sulcus right in the middle, and it divides that frontal lobe from the back, the parietal lobe. And so you have the primary motor cortex, which again is responsible for all of that motor information. So moving away from things like, if you imagine like a bear was chasing you, you sense it from the back. So the primary somatosensory cortex is the back half of that central sulcus. And then in the front half, you've got the motor cortex. In front of that, you have premotor. The very front of the brain is the cerebrum, E-R-U-M. And the cerebrum is important for like memory, thought processing, like personality, kind of all those things go into that front part of the brain. Then you have the Broca's area, which is important for the muscle coordination necessary for producing language. So moving the lips for speech, writing, kind of things with your hands and stuff like that. So primary auditory cortex is right adjacent to that, and then next to that is Wernicke's area. So Wernicke's area is important for the actual name of something, like actual language. And then in the very back, near the occipital lobe, is the primary visual cortex, so that's where you see everything. And again, with the brain, it's important that you remember the right side of your brain controls the left side of your body, and the left side of your brain controls the right side of your body, so they cross over.
So right side of the brain controls the left side?
Of the body. So like my right eye sees, but my left eye processes, left hemisphere of the brain processes what the right eye sees. And so...
do you think he would ask something like, specifically like what does this part of the brain do, or it would be more like application?
I think most of the time it's application, but there is like, if I remember, it's more like knowing what the structure does can help you identify what the answer is, because sometimes he'll throw a bunch of different terms that you might not be familiar with, but if you know general parts of the brain, you should be able to like dissect apart, okay, like this area of the brain is important for, you know, memory or thought processing. This area is like visual, so like at least you can guess kind of what it does if he asks you a question like that. So he didn't make a set of practice questions for this final unit, but I did, and it's like, they're not like, you know, his style of questions, it's more like just content recollection, just to quiz you. And I'll send that kind of closer to the final, probably after break.
Thank you.
So with stroke, it's important that you distinguish the two types of stroke. There's ischemic versus hemorrhagic. So again, ischemic stroke is important because that's an occlusion that blocks blood from getting to a place, and so blood builds up kind of before the clot. Whereas a hemorrhagic stroke is like you're bleeding too much, and so there's blood everywhere. So you can use TPA, the tissue plasminogen activator, to you know, induce the breakage of fibrin clots for ischemic stroke, but you cannot use TPA for the hemorrhagic stroke. Okay. I'm not going to go over this like brain anatomy stuff. That's covered more in the normal neuro lecture. So we'll skip to brain infarction. Basically, if parts of your brain die, then like it's like not really going to be functional anymore, and you can have hemorrhages that cause bleeding everywhere. So let's talk more about dysphagia. So with dysphagia, it's a problem with speech. So depending where in the brain you have that problem, that's what kind of speech problem you'll have. which logically makes sense, right? So if you have a problem with Broca's area, you have difficulty speaking and writing because again, that's the place where you have like information about moving your lips to make a word, like moving your hand to write a word. Wernicke's area, meaningless language, so like people who have damage there, like instead of saying chair, they might say cat. Instead of saying like flag, they might start singing Amazing Grace or something like that. Global dysphagia deals with frontotemporal, so like this front part of the brain. Frontal lobe is here, temporal is here, so it's actually right around here, this connection point. All language is impaired or lost. Just happens to be a very important speech area. Anomia is a problem with the angular gyrus or posterior superior temporal. So that's kind of like around here-ish. And so if you have a problem with that, you are unable to name an object. So anomia is kind of like Wernicke's. Wernicke's is like, it's more towards like, you're saying just like gibberish, like you can't even form any sentence, like it's just like blah. Whereas anomia is more of what I think I just described. Like you called something something else. Transcortical motor, again, is like this portion here, the front part of your brain, impaired output, and then transcortical sensory is like that back portion, and that is impaired input. You can have aneurysms as well. So aneurysms can most commonly occur in that circle of Willis, which is a collection of arteries. And if you have an aneurysm, like this big collection here, it'll increase pressure. When you increase pressure to a certain degree, it could rupture, and then you could have a lot of blood come out, and it's very bad. Yes. It's just like a big, like, lot of it. Yeah, a lot of blood coming out. So you see, it's like a big bulb that forms, right? Big collection of blood there. Arteriovenous malformation. So it's congenital, or it could be if a surgery went bad, and it can rupture if there's like pressure increases on the system. So remember, like, it's bad to have these like arteriovenous formations in the brain, but it's more normal in the skin because you want to have like cooling. So heat cools when you have that direct connection artery to vein in the skin, but not in the brain. Herniation or increased intracranial pressure. So a herniation, hernia is like if something moved into an area it's not supposed to move in. So if we see here, like part of the brain is actually like shifting over a little bit. So it's fair game for him to ask you like any of these types of herniations. So tonsillar herniation is a downward displacement of the brainstem through what's called the tentorial notch. So you see here it's like moving downward. The uncinate herniation is if you have herniation of the temporal lobe into the notch, so the side portion over. Subfalcine herniation is if you have the cingulate gyrus move under the fox cerebri. And then lastly, so those are your three types of things. So fox cerebri is the gyrus closest to the corpus callosum, so it's this area right here. And again, the corpus callosum is what separates left and right hemispheres of the brain. And then fox cerebri is that gyrus, so it's the bump or the fold right next to it. So it is fair game for him to ask you which kind of herniation it is, so like just know like the three different terms. Let's see. Hydrocephalus is if you have too much fluid in the ventricles, and the ventricles, if they have too much fluid, they obviously get bigger. So there's two types, non-communicating versus communicating. So non-communicating is like you made the CSF, cerebrospinal fluid, and remember why we produce cerebrospinal fluid? It's for nutrient and waste exchange, but also two, it's for like protection because it's like shock absorption. If like your brain is moving, then you can kind of cushion the blow by like diffusing the force in the fluid. So that cerebrospinal fluid normally drains through the cerebral aqueduct, right? But if you have a compressed plexus, you can't really drain it out. And then what you'll see clinically is like a kid with like a bigger head but normal intelligence. You won't really see it as much in adults, but if you do, it's not good because a kid, their skull is still growing, so then like with their brain kind of getting bigger with fluid, like at least the skull is expanding, but an adult who has a fixed skull size and a growing brain will have like a lot of headaches. On the other hand, communicating hydrocephalus is like the CSF, it normally gets reabsorbed in those arachnoid granulations, but if it's a problem of reabsorption and there becomes like too much CSF everywhere else, and it's usually seen in older patients, so you'll see these two types of hydrocephuluses in different patient groups, more likely.
What is the communicating?
So communicating has to deal with it's when the flow of the CSF is blocked after it exits the ventricles, whereas non-communicating is like there's an obstruction that blocks it so the CSF can't go out within those ventricles. Yeah. Let's see. So with hydrocephalus, you'll see relentless headaches, decreased mental function. You kind of have like pretty bad balance, things like that, and lots of intracranial pressure, and you'll see it with a brain scan. Trauma is pretty self-explanatory, like if you hit your head, but there's different types of bleeds that can occur. So you can have like an epidural hematoma, so like remember your brain is like surrounded by the pia mater, the arachnoid matter, and the dura mater is that outer space, and the dura mater is closest to the cranium. So an epidermal hematoma or epidural hemorrhage, that is like bleeding that occurs in that outer portion, like between like the very outside of the brain, that connective tissue that's out there with like the cranium, there's bleeding up there. That's typically a cause of an extreme traumatic brain injury, or when you, if you like, for example, broke this middle meningeal artery, that blood can go straight into the dura mater, and if you have a lot of blood there, it's very bad for the brain for pressure reasons, but sometimes what could happen is if there's an epidural hematoma, you could have a herniation during their sleep, the patient would die. So yeah, lots of different things. And then the hematoma can be like kind of in different regions. It could be subdural, it could be epidural. Epidural is typically the more extreme traumatic brain injury, whereas subdural is just like a normal head injury that kind of like hits like kind of closer to the brain. Like that's where the bleeding is. So look, a picture of a traumatic brain injury. Yeah. So you have two protective layers for the brain, the skull and the CSF. And that CSF is, again, to redistribute any shock impacts. If you have problems with the spinal cord, or you can have many different problems with the spinal cord, like one, if their intervertebral discs like kind of bend the wrong way. Two, if they're compressed like up and down vertically. Three, if like a fracture occurs, or for like pretty much like all of these are fair game, but like just know if you have a problem with the spinal cord, like you will have problems with other forms of like sensory and motor processing and relaying of information. So if you have a hemorrhage, it can cause problems eventually leading to hypoxia, because when you bleed out too much, you're like leaving spaces without red blood cells, and they're not really doing so well, and like you have a lot of free radical damage, things like that. He does talk about how like your spinal cord can regrow in theory, but because it's so fragile, typically like something cuts it off and like makes it break again, so then you really won't fix it. So like, within the first 2 weeks it's trying, and in theory it could reach here, but like more likely than not like something will kind of like cut it off at this point, and it won't reconnect properly. So that's why spinal cord damage is so difficult to deal with. You also have degenerative disc diseases. So first, there's a thing called the annulus fibrosis. Think of it as a rubber band, so it's that outer kind of more squishy portion right here in this light blue. But you also have an inner portion that's called the nucleus pulposus. So the nucleus pulposus is more springy, up and down. It's like goopy. And so it's kind of like stretching outwise, like for tension purposes with the annulus fibrosis, whereas the nucleus pulposus is more compression based. Because it's goopy, you can kind of deal with things that like push up and down. And so if you have degenerative disc disease, you have that nucleus pulposus, that goop is like seeping out, and that goop will hit your spinal nerves, and that can cause paralysis when you damage nerves because they're very fragile. The different types of cancers, it's good to know like what the name implies. So like astrocytoma, it's not actually a cancer, but it's more like a lot of the cancers it's like in the brain are difficult, not if they like secrete stuff, but it's more like the space that they're taking away from in the brain, or like the things that they compress on, like so their size really matters. Glioblastoma is a type of astrocytoma. Oligodendroglioma and
They're not cancers?
so these astrocytoma is not a cancer, and I think the other ones are though. These ones,
So glioblastoma is also not a cancer?
glioblastoma is a cancer, but astrocytoma is not a cancer.
He said it's just a tumor of astrocytes.
Yeah,
He characterized both as just tumors. He didn't call them cancers.
yeah. So it's just like a mass that's like in the brain. But I can get like a more formal follow-up on this. There's also medulloblastoma, meningioma, schwannoma, and metastatic cancers. So tumors are bad in the brain because of pressure, and because you have pressure, you have headaches. There's a sign of intracranial pressure called pallidema, and you see it here. It just means that there's like bulging because of that pressure, and you could get bulging in the foramen magnum and near the optic canals. So again, like different cancers or different tumors will form in different areas. Yeah. Let's see. Spinal tumors, so astrocytoma, meningioma, ependyoma, and schwannoma. It's just like, again, showing you a visual, but I don't think he'll ask you like a diagram or anything like that. So like you can just guess that this has to do with meninges, like meningioma. Ependoma deals with like ependymal cells. Schwannoma, Schwann cells. So Schwann cells, remember, they're important for the myelination of axons in the peripheral nervous system, and we'll talk more about that soon.
What was the cells for the
Ependymal cells, those are important for, I believe it's for secreting the CSF. So they make up the choroid plexus, and the choroid plexus within the brain is what produces CSF. And like, remember, CSF drains like kind of like outward, so it first goes through like those lateral ventricles, the third ventricle, fourth ventricle, around the brain, down through the spinal cord to the cauda equina, so like all the way down the spinal cord, and then back up. Bacterial infection, very rare because you have that blood-brain barrier. But what is important to note is that meningitis, like very dangerous disease, and axons don't typically grow there, and it's like just very difficult to treat it. And same with viral infections. It's like once you have a viral infection there, it's like again like how do you get your immune cells or how do you get anything up there? So they're trying to like find different treatments for it.
Spinal cord impingement or something.
So like if there's like something is pressing on your spine. Like,
Is that all of those 3 kinds that will cause spinal cord impingement?
No,
Are you
that was
saying
back
Or
here.
that is.
That's where he talked about spinal cord impingement.
Is that cancer related?
like
I don't think-
this stuff or
Yeah.
this stuff. This.
We talked about it with degenerative disc disease.
I think just this stuff is like, with age, your discs will degenerate, and so the spinal cord just kind of weakens a little bit, and if it weakens to a certain point, then like it might bump into another part of the body. Does that make sense? Prions, they are an important type of disease because they are based from a protein, unlike a virus, which is genetic material, unlike a bacteria, which is an actual living organism. So proteins, again, they are not living, and what they do is like a good protein a good protein, it's in your cell, but this bad protein somehow like gets into your system, and it will bind to your normal protein and cause a conformational change, which will convert it into like a bad, unusable form, and then it starts to aggregate, and then these buildup of like proteins that are not good can infect different parts and accumulate in like your brain, for example. So mad cow disease, Creutzfeldt-Jakob disease, also Kuru is another disease that he mentioned, but I think that's it for the 3 kind of prion things to know. So mad cow, Creutzfeldt-Jakob, and Kuru. Kuru is like when like I think there was like indigenous tribes or something that like would eat like flesh, and then like that's how they would contract the disease. So yeah. Degeneration. So there's all these different types of diseases. Like let's go through them. So Alzheimer's. I think we all know what that is, but it's a very specific form of dementia. So there's different types of dementia or layers to it. It's like you can have anterior grade amnesia where your like older memories, but you can't form new ones. So his example was like, can you play Scrabble? Like, yes, but you can't play a new game called Mexican Train Station. And then retrograde amnesia is kind of like the opposite. So like you could form newer memories, but you can't keep the old ones, and it's much more rare. Alzheimer's could have both forms of dementia. Yes. So senile dementia is more with this anterior grade, so it's like you remember like older things. So what happens in Alzheimer's, you have these alpha beta portions or peptides in a certain structure, it's a transmembrane protein that like it typically gets cleaved and broken up and like processed well, but because it's not really getting cleaved in the Alzheimer's condition, then you have aggregates that form, and that will cause these amyloid fibrils to form. And so I remember like last year, the year before, like Georgetown found a way that like they can detect Alzheimer's like before patients die, or like they can officially diagnose it or something like that. I didn't read the article, but like typically what they used to have to do is like wait until after someone died, check their blood, and like see whether there are these aggregates, like to confirm.
They cause what aggregates where?
This is like in the blood. Yes, I think it's like brain.
I don't know.
Brain. And so you have you're losing blood flow to the brain, and neurons are dying because there's like all these plaques that are forming. So these
Is that Is plaques the acronyms?
Fibrils, yeah. So different word, but yeah. Amyloid, fibrils, amyloid plaques, same word. Just stuff is building up in the brain. Because it's building up in the brain, then you start to damage other things. You damage neurons, and you lose blood flow. Major motor system diseases and disorders. I took this and threw it on my cheat sheet. I don't know how helpful it was, but it was just like, here's a lot of information. Maybe it might be useful somehow. Yeah, I'm not gonna read through it, but you can, for your benefit if you want. I don't know. Parkinson's is a problem with the substantia nigra. And so remember, substantia nigra is part of the, I think it's the tegmentum of the midbrain. So you have two parts of the midbrain of the tegmentum, which is the red nucleus and the substantia nigra. And so substantia nigra, this area, what happens is the neurons, they typically will make dopamine from this area, and dopamine is what gives you like reward, and it's important for like inhibitory stuff. So like if you have so the substantia nigra in normal patients will inhibit unconscious movements, but then like in Parkinson's patients, because it's damaged, then you have like the hand tremor, or you have other problems start to accumulate. So you can move from one thing to another, but you can't stop once you start moving. So dysdiadochokinesis.
Isn't there like an acronym or something for that?
PD?
No, not I don't know what I mean.
Okay.
It's like a shortcut to remember what Parkinson people do. Something like ship or shit.
Okay, thanks.
So the neuron substantia nigra, like, did that destroy the neuron?
So substantia nigra is normal. It's a normal brain structure. In Parkinson's patients, they have damaged or dysfunctional substantia nigra, so you cannot produce dopamine that well. And I think their substantia nigra, it's like nigra meaning like black, it's like less black, and there's like lower levels of this thing, neuromelanin, so it like looks less black.
What is a Louis thing?
Lewy bodies, forgot what exactly they do, but that word is associated with Parkinson's. Are you Googling it? Do you see it in your notes?
Yeah, I see in the notes. It said Louis, but
Lewy body and a neuron from the substantia nigra steams me. Oh, yeah.
Okay, cool.
So probably not.
Well then you probably don't need to know it.
I think that's all I said, just right there.
Okay, cool.
Associated.
Association. Yeah.
no, I found the mnemonics. So if anyone wants to
You
know,
want to just say it?
it's called TRAPS.
What does it mean?
Tremor, rigidity, akinesia, postural changes, and stare. That's what Parkinson people would do. They would like shake and they like walk like that. They can't stop.
Like, in my notes, it says like problem with, well, I like wrote like there was a problem with the basal ganglia for
Mhm.
Parkinson's. Is that
We
like
talked about that.
is that like the same thing? Is the basal ganglia the same thing as like similar to substantia nigra, or like is that like different? Or?
He said that it was responsible for starting and stopping actions. Is it?
So the basal ganglia was like, if you guys remember the human bio, there's like that collection of like, the putamen, the amygdala, like blah 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 blah, like caudate nucleus,
The
those six
substantia
structures.
nigra part of it.
I don't think so, but it's like next to it.
So they're both like they both have problems, I guess, in Parkinson's.
I think so.
Is that what
But yeah, like Huntington's disease is a problem with the basal ganglia, is what I have written here. It's a problem with the caudate and the putamen.
Substantia nigra is in
So this is a damage in a different part of the brain, caudate and the putamen. So you have excessive output in this case, so it's always twitchy, jerky motions. Multiple sclerosis is a problem with 4 different types of how it manifests itself. You can have relapse and remittance, RR, getting worse, getting better, like one after the other, kind of. Progressive relapsing, so just getting a lot worse. Primary progressing, so it gets steadily worse with attacks. And then secondary progressing is relapse, remitting, and then you have primary progression. So it starts off as RR, so gets so bad, then gets better, but then it becomes progressively worse. So MS is a problem with demyelination, and so you'll see demyelinating disease of the CNS, central nervous system. So the immune system attacks oligodendrocytes, and oligodendrocytes were the cells responsible for myelinating axons in the CNS. And when you have demyelination, do you guys remember, it's a slow signal, so then you can't process things as well. And the clinical manifestation, you'll see a right leg weakness, for example, that's not bilateral, and you can't find a physical damage to it. And most people are diagnosed with more a sensory problem or a muscle weakness rather than MS. Let's see. This slide has to do with like how you can have demyelination. So it could be segmental demyelination, right? And that's randomly interspersed, like how you're demyelinated, versus axonal degeneration, seen with polio, is where like the entire axon just kind of like went away. Axon and its myelin sheath undergo anterior grade degeneration, so you don't see the nerve anymore, and then you'll have atrophy for anything kind of downstream of that, because it won't be used anymore. So Guillain-Barré syndrome is an autoimmune disease of the peripheral myelinating cells, so that's the PNS. And then Schwann cells Guillain-Barré is for peripheral myelinating cells. Schwann cells are the cells that myelinate the those regions in the periphery, and so they form this onion bulb kind of like neuropathy, so they look like dark circles. So Guillain-Barré is for PNS problem with the myelination, and then the MS is
Onion rings on Schwann cells or what?
yes, attacking Schwann cells, which are responsible for the myelination. So going on to muscles. Let's see how much time we have. All right, cool, halfway done. So with muscles, because he didn't teach it to our unit, our year, I don't know
So we
what
teach you.
questions he has on the exam about it. Okay, that's my caveat for all of this. So a muscle you can break down. Remember, muscle cells are a little bit different than your normal cells because one muscle cell is an entire stretch of multiple components. So one muscle is consistent of muscle fibers. Those muscle fibers are the individual cell. So they're all multinucleated, they run the length, so it's this entire long thing. Within each fiber, you have myofibrils, so smaller fibers, basically, and within each myofibril, it can be subdivided into sarcomeric units. So one sarcomere has an I band, think light, and a dark band, the A band is dark. And so nuclei are all throughout this cell, this muscle, because like it's so long, it's so big that like you have to kind of keep it up, like all the functions of a nucleus. So you have an M band, that's where the myosin is attaching, right in the middle of the dark A band. and then you have a Z disc in the middle of the light actin I band. Okay, so light, think actin, dark, myosin. And so when you have a contraction of these sarcomeres, you'll see compression and stretching relative to like a reference frame. And so it's like I and A, they're kind of like moving around, and that's how you get muscle contractions. This is a couple slides taken from Human Bio when JP taught it to us. So it's like, you can read through this on your own. But actin is like beads on a string, and so it has myosin binding sites. So actin is like this long stretch. Myosin is what's actually going to move and pull the actin. So you have these voltage-gated calcium channels that when they're triggered for release, they will start to dump acetylcholine. So acetylcholine is the neurotransmitter responsible for activating a muscle potential. So calcium comes in, you release acetylcholine, acetylcholine binds to receptor, and so sodium will now rush into the cell. Once sodium rushes into the cell becomes depolarized, and when the cell becomes depolarized, this voltage-gated calcium channel inside the cell will open up, which can act on the sarcomere, sarcoplasmic reticulum, to start releasing more calcium. And then all of this calcium inside the cell will trigger a muscle action potential. And so the way a muscle action potential works is typically, or normally under normal conditions, you have your actin, which is like this like long line, and it's typically bound by troponin or tropomyosin. And so troponin, it will bind on the myosin binding sites, but when calcium comes in, it will tropomyosin is binding the myosin binding sites. When calcium is released, it will bind to troponin. Troponin can then move tropomyosin off the myosin binding sites, and then you expose those myosin binding sites. Once you expose the myosin binding sites, myosin will bind onto actin, and then you dissociate an ADP molecule and you pull. So each muscle contraction pulls an actin like a little bit farther along the track. But you're stuck in a fully contracted position, like you've pulled, but you can't start over until you have ATP, so energy. So energy has to be used to break off and release the actin from myosin. And then you repeat the cycle. So starting over again, it's like you have the muscle action potential occurring, so like all the stuff we just said, like with the acetylcholine activating, so the electrical signal travels down T-tubule, so a muscle action potential, like you have acetylcholine, it gets bound. Once it gets bound, you have signal goes down, right? This muscle action potential travels down T-tubules and is recognized by the sarcoplasmic reticulum within each of these myofibrils, or around the myofibrils. Then they release calcium by the method we just discussed, and then you have this whole circle. So the circle continues to repeat down here, like from ATP hydrolyzation to ADP, myosin can bind onto actin, ADP gets off, you have a power stroke, and so you pull actin farther, you use energy, and you break energy to break that bond between myosin and actin, and you can start all over again.
I have a question. So, like, the ADP, what is it?
It's just like a molecule that's just sitting on there,
Okay.
and it's not really going to do anything. And so, like, once ADP falls off, that's when the power stroke occurs, and you can pull actin a little bit farther. Does that make sense?
So, if you pull actin farther.
That's the
that,
muscle contraction.
that lets the myosin bind to the actin. Is that
So the myosin wants to bind to actin to move it, and you only move it when you have calcium in the system.
right?
And calcium is released in the system because of the muscle action potential. So acetylcholine getting released, telling the cell to like allow for all this to happen.
Okay, so then, how does ATP form then?
ATP is just in your system, like mitochondria, right? And so ATP is floating around, and once ATP is used, like it binds to this like empty site on the myosin, then you can break ATP into ADP plus the phosphate group. The ADP stays on the myosin, and then like you finish the circle. Okay. Yeah. This is like the same thing, just like in different words. I'm skipping it. There's different muscle fiber types. You have a slow oxidative, that's type 1. You have a fast oxidative or fast glycolytic, type 2A and type 2B for muscle content. And so oxidative, slow oxidative type 1 tissue is important because you can have high oxidation. Like, what does that mean? Like, it means you can do very well to be indefatigable. So you are able to constantly generate more energy in the long term. So you have many mitochondria to generate energy for the long run. And it's also a red fiber. Whereas fast glycolytic or fast oxidative tissue type 2 and fast glycolytic tissue type 2B, those are important because they contract very quickly, and that's like if you're sprinting. But to use that much energy, you don't have as much oxidative potential because like you need the energy right now. So you use up all of your energy up front, and then you get tired. Okay, so red muscle, slow oxidative, slow twitch type 1 muscle is kind of like for the long run, whereas white muscle, fast glycolytic, or fast twitch deal with type 2 muscle. So type 1 slow twitch has all of these different components. Low contractile force. So like you can lift something, but you don't like use all of your energy to lift something all at once. It's kind of like gradually, right? And then it's like for long term. Whereas a slow twitch, fast glycolytic tissue will be high contractile force because you're about to sprint. And so it has relatively less mitochondria, less energy, very easily fatigued. Okay. And then motor units, just to talk about those, like you have a single neuron and it controls multiple muscle fibers. You control less fibers if you want to dedicate more like fine control over it. So like the eye, you want a lot of control, so you have less fibers, right? And then the efferent neuron for the quadriceps, for example, a large muscle that doesn't really do much specifics besides like standing, moving, that kind of stuff, that's like a lot of fibers because like you don't really care as much. Like it's not as fine control. So when you have muscle action potentials occurring, you recruit different motor units to do different things at different times. And you can have like multiple stimuli temporally, so like you can tell one single motor unit over and over again to do something, or you have multiple motor units that's spatially like summing up all of their information. You don't need to know that. Type 2 fibers get called on first. Type 1 is later because it'll like last you in the long run. Type 2 is all for the beginning. Yeah. But in terms of like what makes up most of the body, it's like the type 2 stuff. Okay. Alright, so kind of like finish strong here, guys.
Yeah.
You have normal muscle reinnervation, atrophy, just like, atrophy can happen. Duchenne's muscular dystrophy is a problem because like typically to build muscle you break it down, and then when the body like rebuilds new muscle in the break time that you're relaxing, you build stronger new muscle. In Duchenne's muscular dystrophy, every time they break a muscle, they can't fix it. So basically it's like they have broken muscle and it's like not even like repaired. So it gets very, very tired, lots and lots of muscle damage, very, very fatigued.
What is Duchenne muscle?
I just said. So it's like a condition where you can't build back the muscle.
Old people.
I don't know what the treatment is. It's
I was just gonna say like don't move.
yeah.
Because that's kind of what he made it sound like. He's like,
Well, he
if
said-
they move, they're
Yeah, like someone asked like how do these people like die, and he said like they're just from fatigue of like their breathing and like swallowing muscles and things like that.
Yeah.
So
I don't know
there's
if
no
there's
treatment?
I don't
Is it only for old people? What causes it?
no, he said because he said like it's the roads don't have lots of it. He said it's something gets. This person does it before. Oh wait, no, it's autosomal.
it's an autosomal dominant disorder, I wanna say.
X.
Yeah, so
Excellent.
it's
Yeah,
genetic. Okay. Alright, so with this slide it's just showing that you have this molecule called dystrophin that's like important to like it's kinda like you have a muscle like fiber, right? Or like the plasma membrane within that muscle, and it's like when you have force on it, you try to distribute the force evenly all the way across that plasma membrane. And then I think okay, so mutations in dystrophin are associated with X-linked muscular dystrophies. Yeah. So a problem with the dystrophin here, I guess it could be like you don't properly like distribute the force around. I'm not sure how much detail he went into on this.
He was talking pretty well.
Okay, well then.
It was pretty straightforward.
Cool. Alright, this slide was just showing you like a stain for dystrophin, and it's just showing you how in the Duchenne's muscular dystrophy you don't see the same type of staining, right? So you don't have dystrophin, you can't really like build your muscles back, and you see a lot of creatine kinase because like muscles are getting broken down, and like yeah, your neutrophils can't really like clean, or they come in to clean up all of that mess. So you see the dots here because it's like you have like broken down muscle, and it's like, oh well, it's not functional, so you got to clear it out. Myasthenia gravis. There's two things that might get confusing. So myasthenia gravis is like I blocked the receptor on the muscle for acetylcholine. Myasthenic syndrome is I blocked the actual channel that releases like the pre kind of portion. So it's like what's releasing the calcium, and so we can't break, we can't like initiate, versus like myasthenia gravis, we can't recognize that it's there. So clinically how that happens is like myasthenia gravis patients will have weakness in the evening more than the morning, because like you have like an accumulation of acetylcholine like overnight, and then like all that acetylcholine can kind of like find the free receptors like in the morning, but then by the end of the night like you kind of like used all of your free acetylcholine. Versus a myasthenic syndrome is like you can't really even get rid of acetylcholine, it's like not even getting released, so you can't really like fix it. So then you don't get better from morning to evening. There's many different impairments of the neuromuscular junction. You have a blockade, neuromuscular blockade by like poison curare. And so curare is like if you blocked the actual acetylcholine receptor. Clostridium botulinum, Botox, it prevents the muscles by preventing acetylcholine from dumping out from their vesicles, and then Clostridium tetani is like you have too much neurotransmitter, so you're so stimulated that like you have like locked jaw, for example. Osteocyte builds muscle, or like they are the muscle. Osteoblast builds the muscle. Osteoclast eats it, breaks it down. Fibroblasts, they lay down tendons, ligaments. Chondrocytes are for cartilage. Osteocytes, bones. Let's see. You have cortical bone, which is called compact bone, medullary bone, which is called spongy bone, or trabeculary bone. So the compact cortical bone is kind of like the outer parts, and then you have spongy bone towards like the ends. And if you need calcium, you can break down bone. Remember back to like parathyroid hormone. PTH. Many different cells of the bone, many different disorders of the skeletal system. Read through it. Kyphosis is curvature of the upper back. Lordosis is lower back. Scoliosis is an S kind of shape, so it's lateral, and it tends to hit teenage girls more than boys. Osteoporosis is like bone demineralizing, and so it's like very much broken down compared to healthy bone. You can have osteopenia, which is like the in-between normal and osteoporosis. Osteoporosis can be due to age, and then it could be due to like disease and stuff, but like basically it's like kind of like different bone loss, and it can be in the spongy or the cortical bone. This picture is showing kyphosis, so curvature of the upper back. Paget's disease is a rare disease when the bone remodels too quickly. Could be because of a bone virus where immune system is attacking bone. It's a rare, very rare disease. Prevalence about 3% in people over age 40, and yeah, I think it's basically like you're breaking down bones, or like you're losing bones in the skull. That's pretty much it. Hyperparathyroidism, because remember, parathyroid can take away from the bone to like boost calcium or like deal with calcium release. So too much parathyroid can break down too much bone. Osteomyelitis, so inflammation of the core of the bone, so inflammation here. Osteomyelitis again, it can kind of like eat through that middle portion of the bone, and then like bone and joint tumors are.
So does it fix?
Does it fix itself? I don't think so, unless like you cut it off early enough. I think it's like once you lose it, it's probably not gonna build very easily. Or take a very long time. You probably need like growth factor or something like that, because like bone is like a lot of like those stem cells, like hematopoietic stem cells. So bone and joint tumors, mostly metastatic, and so they kind of just like branch out everywhere. Joint disorders, you have two types of arthritis or joint diseases. You have non-inflammatory and inflammatory. So the non-inflammatory would be like osteoarthritis, so it does not have inflammation associated, but it's similar to inflammatory conditions because like it hurts. It's like joints, dislocations, things like that. Inflammatory joint disease, like the immune system. Rheumatoid arthritis is autoimmune disease. Gouty arthritis, diet-induced like uric acid buildup, and then infectious arthritis like Lyme disease. Osteoarthritis, again, not inflammatory, so it's bone to bone. You're breaking down like that, like cartilage that's in between, and then so you have like damage, joint pain. You affect many different regions of the bone that have that are like commonly used and have a lot of pressure. Rheumatoid arthritis, on the other hand, is when the immune system attacks joints, and you can have an ulcer, so your fingers kind of like branch apart weirdly. Let's see. Gout is when you have a buildup of uric acid, and then it's lots of pain, like in the feet, for example. Tophus is a white crystallized uric acid, and it chews away at the joints. The problem with the joints. This is a slide going over the actual synthesis, metabolism, and elimination of uric acid. You typically get rid of it in the kidneys or intestines. Thanks. Lastly, okay then, cool. I
So,
guess we
sorry,
are.
I just have a question. Like, what
Yeah.
is the difference between the gravis, this, and
Myasthenia gravis and syndrome.
Yeah.
So, gravis is breaking like the postsynaptic receptors. Syndrome affects the presynaptic, so you can't even release the acetylcholine across the synapse. Whereas gravis is like you have acetylcholine floating around, but because you block the receptor, it cannot activate downstream. So that's why in the morning, if you have so many receptors, we're almost done. If you have so many receptors that like, it's so many receptors that are blocked, but yet so much like accumulated acetylcholine in the system, based on like sheer number, you could have activation in the morning.
You could have activation in the morning for gravis.
Yes.
What kind of activation?
Well, like
You don't want
just
that activation?
no, like muscle function. It like functions in the morning better.
Do you